All right. So today's talk we have Alexander Crimi. He's a group leader for the Brain and More Lab at the Center for Computational Medicine in Krakow, Poland. So Alex, he's a traveler. I would say that he's done his studies in Italy. He's been in Switzerland, in the US. He's studied in US. He's worked in US and so many other countries. And finally, has a group and is leading a group which is developing neuroimaging technique or the development in Krakow, Poland. Alex has been visiting Ghana for several years, actually, much more than I have even been teaching in Ghana, honestly. So he's been coming to the African mathematical sciences in Ghana. I used to be in Ghana, and he's been going there. He's been coming there, visiting almost every year for several years, teaching some of the things that he's going to talk about today. He's been teaching Ghanaians, not just Ghanaians, but a lot of African students who come to AIMS to study. He's been teaching and supervising as well there. And I think you even have a Ghanaian research student in your lab at the moment. And he's an author of a very new book, which I'm sure he will show, or he will at least talk briefly about it as well. His main interest is in medical imaging, particularly neuroimaging, and interested in prediction of diseases such as glioma, Alzheimer, multiple sclerosis, and so on. He's really interested in not just the academic aspect of it. This is something we share in common, not just academic, but really innovations, trying to be an entrepreneur with academic ideas. And this, I'm sure he will talk about it briefly in his presentation as well. And yeah, he teaches, one of the things he teaches in machine learning in medicine, or machine learning for neuroimaging and neuroscience. I'm not going to bore you a lot. I'll just leave Alex to just give his talk. Alexo, you have 1 hour to do your talk, and then after we can engage and ask questions, and we see how we go about it. So please, you're welcome to make a presentation.
Okay, thank you for the great introduction. Can I try to share my screen?
Yes, go ahead and share your screen.
Okay. Can you see it?
Yes, I can see your screen.
Okay, then oh, no. Yeah. Okay, then as just a brief summary, I also to explain what is the institute. So I'm actually have two positions. So one is the university, where actually I have a title there, but I don't have a lab. So I'm a professor at the AGH Technical University of Krakow, but I'm more just a lecturer. So I just do teaching and some thesis. While I'm a group leader at SANO, that's the Center of Computational Medicine in Krakow, which is like an independent institute. It's still within physically the university, but it's like an independent thing. It's like let's say AIMS and University of Cape Coast. Like you have these two structures, something like that. So okay, I have a long presentation, but feel free to interrupt me many times. Like, don't be scared to be too polite. We are all friends. Actually, knowledge is just interaction. So anytime you want to stop me, I don't know, raise the hand and I will stop. Or if I don't see that you raise the hand, just even speak directly. So I show you how it looks like. So this is Sano. So we are in this strange yellow place with the yellow windows. It actually looks nice from outside. From the inside, it's a bit strange. And you see on the left the current groups. So we are most of from different countries, and yeah, for some time we had some interaction also with some people from Ames, but then right now one left. So it's already in the Indiana University in US, and another one was kind of working remotely from Ghana, and she's now a PhD student in US. So you see only Obruni, but there has been some other people in the meantime. So and I wanted to I recovered some pictures. So I want to show you. You heard it, but I've been to Ghana other times, and on the first picture is like the aims, the first times, actually the first year that aim was created. And then also we did some project in Anomabo and other village nearby. And also we had some funny things like claiming that we were doing science inside the trotro. So it's a funny picture while we were reading papers. So just some memories I had of Ghana. But starting more on the presentation today, no, the presentation will be mostly about science, like what we do. But maybe in the end, I have some discussions maybe on how to do things going outside academia. As I was telling you, don't be too much attached to stay in academia. Well, maybe actually a good way that we can make all improve the development of our own country. It can be Italy, Poland, Ghana, or wherever, is actually that you create your own company. You try to do something in the real world, or even if it's not in the real, like making a company might be doing some applied things. Let's say deploying what you're doing in an hospital, so not just being on closing at the university. So I made this summary like of how I imagine that there is this loop of people just that publishing papers and then finding grants and hiring other scientists, or you can go in the other arrow, the other direction, where you create your own company. I will come back more in the end, at the end of this. So I will start now a bit of with the science. So what we do, then I will we in our group we are mostly interested in representation of the brain like this, with this graph. I will explain you how we create this. And we are also interested to relate this kind of representation of the brain that's called connectome to other aspects. Let's say either some uh, practical disability or disease, or maybe we go in the opposite direction where we try to correlate these connections to something related to genetics, genomics, or other omics. Of course, like everybody, we many people are doing, and also probably is very hot at the moment, is that we use AI, artificial networks, for example, machine learning, which actually many things were inspired by the actual physical brain. And like in a way, actually now we use it to understand the brain. So it's like going back to the origin. Like these things were introduced copying some mechanism of the brain, but now actually we use them to find out or classify or make some kind of diagnostics for brain disease. When I say like classify, is like we generally what we do is that we collect two groups of data. Let's say we have some connectomes of people, let's say with Alzheimer or schizophrenia or any other disease, and then we have another groups of other connectomes like this from a healthy subject, and we try to find something either that we can make it's in the method that we can automate some kind of classification and help doctors, or maybe we go and look what are the differences, so that we can actually give new insight to the doctor. We will give more new biomarkers, so that they can also evolve in there, or we give them new insights. So I will go super quickly, just telling you how we construct this graph, because there is not only one way. There are several ways to construct this graph. In fact, when we say connectome or connectivity, there can be something coming from a structural anatomical connectivity or functional connectivity. There are others, so that are a bit more complicated, but I will just mention them quickly. I will instead try to explain you more the first two. And as I was telling you, this for us, this connectome in the end, do you see my mouse? The arrow of my mouse?
Yeah.
Yeah, I can see it. Yes.
So what we do in the end, they look very complicated, but in the end, for us, they are like features to discriminate something. Let's say someone class A is a disease and class B is someone healthy. So we use them as a very complicated features to just make kind of the discriminations. It's just that they show you something specific that is supposed to help us to do the classification. I start from the structural connectivity. So the idea, probably that in the brain we have let's call white matter, although it's kind of a light gray, or and we have the gray matter. So this that is actually the dark gray part of the brain. And actually, the reason why it's white is because we have the axons. So this long extension of the neuron is in the white part, while the center of the neuron are in the gray part here. And the peculiar thing is that is and this generally, when you have water molecules are moving freely in an environment. But if they are like bounded by the axon, they are forced to move along this main line. And the fact that it's forced to move along this line is like constructing some kind of direction to the water. This thing is actually very important because I lost the slide, but this thing is very important because we can somehow, if we use magnetic resonance imaging, you probably seen when you go in the hospital, is this big machine with the magnet. We have some ways that we like recover the magnetic field from different angles, and we are able to reconstruct this direction. And the fact that we are able to reconstruct this direction substantially is allowing us to build this kind of nice tracks. You see all these nice lines. Okay, this was from a very old work we did it with mice. So these are not human brains, but I can show you more other things that we did with the brain, with the human brain. These are substantially allowing us to reconstruct the direction of this. So we don't see the entire all the neuron, but we can see the axon. So what we see here is we are practically reconstructing only this part, which is actually important, because in this way we don't just look at the brain like you see here, like kind of white matter, gray matter, but we start seeing some kind of connections. And when you see these colors are not random. So of course in your brain they don't exist. It's more like a graphical representation that we use. It's they are like supposed to represent, let's say, red is going from this direction to this direction, and blue from this direction to this direction. And of course I cannot show you, but green means like the other direction. And long time ago we did it something that we were using like some kind of sparse discriminant analysis to figure out what were, let's say, the features that were helping us discriminating this group of mice. So we had some mice with autism and mice that they were like normal, and we're trying to find out the difference. This looks colorful, but it's not very practical. And okay, here you see it's not moving. Oh, sorry. It's not very practical because now this is from the human brain. It's not very practical, but you have to imagine that for the human brain we can do the same, but it still looks like spaghetti. It's like a big bowl of pasta. It's very difficult. Maybe if you're a brain surgeon, it's easy. But if you want to automate this, if you want to feed this into some kind of machine learning framework, is not very practical. So what is the meaning of this structural connectivity? Is like we take these spaghetti neurons coming from the magnetic resonance machine, and what we do is like we have some way that from known anatomy of how the brain is organized, and what we do is like all the tracks going from, let's say, from this red region to the green region, instead of looking at the spaghetti going in this direction, we just put one edge going from the red to the green and one node per region. And in the end, if you do this for the entire brain-wide, you will end up in something like this. So and of course, you can also, like, depending how many fibers are going from let's say the red to the green, you can decide the stroke of the edge. And like we used to do, like in graph theory in complex networks, for example, one node, I was telling you, represent one area. Depending how many fibers are entering, we can establish the size of the node. So you probably can see some nodes are smaller, some are bigger. That's because one of these region is probably having more fibers entering. So far, so good? Are you okay with this? I think.
Yeah.
Okay, because I'm just telling you the features. We are not yet using machine. This is just features. And then there is another features that is called functional connectivity, where instead of looking at the structure, we are looking of activity. What does it mean, activity? Is that also again using magnetic resonance imaging, like instead of, it has been observed that you cannot not only look at things like given the structure, but if you acquire if you take many, let's say, pictures in 3D of the MRI scan, the signal is changing in time. Why it's changing a bit? Because depending of the activity of a specific region of the brain, you have more blood or less blood, and this is something that the machine can detect. Of course, these are subtle changes, but they can still be detected. So here, if you see in this picture on the left, let's say I'm considering only this voxel. So voxel is just a pixel in 3D. If you consider this pixel, this voxel in time, you can see that in time the signal is changing. Like it started being higher and then is less. This is probably because there is less blood, more blood at the beginning and less blood. You have to imagine, like, if you are doing something, let's say, I don't know, holding, doing something with your hands and then stopping. Maybe in the region that is related to the hand movement, there is a lot of blood, and then I stop doing with my hand, so the blood is less. It needs to be less blood. So imagine again, if you do this brain-wise, you have practically for each part of the brain, you have all this time. So we were saying that there is this functional connectivity that the signal is changing in time. Now, if for example, I'm looking at these 3 areas. So okay, you don't need. I mean, this is not the neurology course. You don't need to know what these 3 areas means, like media prefrontal cortex and cingulate jars, whatever. So imagine these 2 region, like in time. Do you see something interesting about these 3 regions? So, like the orange is the media prefrontal cortex. The yellow is the posterior cingulate gyrus. Do you see anything interesting? Nobody wants to guess? Or you're still like shocked?
Yeah, I think we're all shocked. Yeah.
Okay, maybe I help you because I'm also a bit shocked. So if you see, so the one
Yeah,
region is like
but I mean, maybe you can ask
yeah.
the question again.
So I was telling you that here in this graph on the right, there are three time series from 3 specific regions. I was asking you if you see something particular in these 3 time series. Because yellow is this area here, orange is this area here, and the blue is this area here in time. Okay, maybe I help you a bit. So I don't know if you see, but kind of the red and the red area and the yellow, they kind of move together. Of course, not exactly overlapping, but they move together, and the blue one is going the opposite. So what this means is that sometimes in the brain, some areas are like coordinating together. They are like correlating. And this can be another way to construct the connectome, in the sense if one region and another region are the brain signal is correlating, you put an edge, or if it's not correlating, you don't put anything. This is something what we call functional connectivity. Of course, again, like the other one, we look at the overall signal in one specific region to another region, but it's substantially because another way of seeing the activity in the brain. In the end, this is still the feature. We still haven't used any AI on this, but this is still how one way that we can use to build this graph. Where is it that I was showing you here? So why I was telling you this is because you have to imagine you have this kind of graphs for, as I was telling you at the beginning, that you can have a lot of this graph from one disease and a lot of this graph for healthy control. And now if you have been able to construct them like this, then you can take the, let's say, the adjacency matrix of one brain, and you put it in a just in a something like you put it like in a long vector, and then you can pass it to some machine learning, like support vector machine or other convolutional neural networks or something. These are your features, and you can use them to classify groups of, let's say, people with Alzheimer or from people without Alzheimer, like that you can make this kind of automatic distinction in your framework. So we did it. This is something work we did some years ago, where we constructed these kind of features like this, but not for Alzheimer, but for autism. And we use some kind of classifier that's called support vector machine, and we were actually able to classify with a kind of a good accuracy people from with autism from people without autism. And what I'm plotting here, these were the let's say the features that were more discriminant, that were more relevant. So I'm assuming you're familiar with support vector machine, or it's something new.
Well, some of the people online is new. Some of them also they know about it.
So with I say it super quickly. So just so the support vector machine is, you consider you have like your feature space. Here the feature space is just one dot. In our case, the feature is the entire adjacency matrix, so it's higher dimensions, but the idea is the same. We try to find an hyperplane that is separating two groups. And yes, essentially with the support vector machine, you try to tune it to train this hyperplane separating the space with some kind of vectors holding this hyperplane. And it's one machine learning model that, of course, there are many. It's very old, and there are many other similar classifier like linear discriminant analysis. But what was interesting for us is that we were using this complicated representation to classify the two groups, and also since thanks to the way support machine is built, we were able to going back to see what were the vector features that were allowing us this discrimination. So we were using it to find these relevant edges. Sorry. And okay, now this is another thing that we did that is not much machine learning, but I want to mention also because it's from a former student from Ames that she did a PhD in Cape Town. What she did was that we were having some this data from Alzheimer, and we were like comparing them in if there was some changes of the graph in time, in the sense like if you have healthy people, they are probably, even if they are 80s, even if they're old, the connections are not that changing that much. But if they have Alzheimer, probably already after 2 years, because of the disease, there start being more disconnections. So some of these connections are disappearing. So the brain is changing. This graph is changing. This was like some of the elements of her PhD, and she investigated these, and she confirmed that the connectome of people with Alzheimer was changing in time. So she shows some statistical significance. I think this was just a t-test for some summary statistics of the graph. But what was interesting is that then she applied some bioinformatics that she's was working on to see if some genomic data that we had from these subjects were able to correlate to these changes. And actually she found something interesting, because we found four genes from the genome that were like positively correlating with these changes in the brain connectivity. So I don't know how many of you is doing a genome-wide association study, but I share that for those who are doing bioinformatics, it's possible to correlate genomic data to this representation. Still, there is no machine learning, fancy machine learning. I will start with the machine learning part soon. Actually, now. So we most recently we did another project still with related to Alzheimer. Because essentially what happens with Alzheimer is that it's a neurodegenerative disease, like that people start, it's very debilitating. People start losing memory, start not functioning very well, so the brain is deteriorating. And why is happening this? It's because there are some misfolded proteins that they start accumulating in one part of the brain and they slowly spreading. At some point they cause the death of the neurons, so they practically destroy the brain. What is interesting is that the spread of this protein, it's similar to the spread of other diseases. So I will come back in the next slide more. What I mean is that you have to imagine that, let's say, one part of the brain is, let's say, someone with COVID or whatever, maybe some communicable disease, and then of course they are connected to another part of the brain, so they start infecting the other part of the brain and slowly it's spreading. So this you can imagine similar to COVID or tuberculosis, like you start saving something and it starts spreading. You have to imagine, like for COVID, let's say each node will be one person. In the brain is one brain region. So what we were able to do is that there is not magnetic resonance imaging, is another modality called positron emission tomography that allows you to see how many of these misfolded protein there are. So what we were interested is that given these initial features, so how many and where there were this protein, if we were able to predict after 2 years where this protein will be. So what is interesting is that we were not the first investigating this. So there were two state-of-art model. One that is coming more from physics, from the world of simulation, because there is a way to define like the it equation, like with partial differential equation, that um, you practically model like the brain is like a finite element. All these nodes are like finite elements, and you are simulating like in it equation, you're simulating how the it is spreading, but instead of simulating the it, you are simulating these misfolded proteins that are causing problems.
Yeah.
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 And another way instead is coming more from epidemiology. So there is a guy who's called Iturria Medina, who took one model used from epidemic spreading for infectious disease and did exactly what I was telling you. So he modeled the brain as if one node is one person. Let's say one guy has the COVID and he's passing COVID to the other guy, then he's passing COVID to the, and then there is, in the end, there is a spread of COVID. Of course, he was considering Alzheimer's, so this misfolded protein was not considering COVID, but this was his idea. What we did instead, we approached this problem with the more machine learning style. Okay, first we did something with not was not even machine learning. It was more coming from the world of statistics. So it was we used something called autoregressive model, which is, well, I wrote this this equation here so that we have like y t minus 0.1. So at the previous time point, we had some values, let's say the initial protein deposition, and we were trying to predict at the other time point the spread. And we needed to find these coefficients in the brain that represent this prediction. And another thing that we investigated is called graph convolutional neural networks, which is similar to the traditional convolutional neural networks, but is more it's something more specific for graph, for networks. So we investigated these two more statistics or machine learning approaches, and we tried to compare to these other two approaches that they were more related to, as I was telling, one is more from physics and one is more from epidemiology. And we compare all four. And what we noticed, okay, first of all, that actually the partial differential equation and the epidemiology, they don't work that bad. We were actually achieving more or less the same thing. The one that worked not so good was actually the graph convolutional networks. And the reason is because we are using high dimensional representation. So when we are passing this in the graph convolutional networks, there are too many nodes, too many features in the brain. And also we have very few, we use very little subjects to train. So these traditional graph convolutional networks didn't perform very well. So here I'm representing the error, and the one with the highest error were the graph convolutional networks, while this is the error of the other methods. You see they are more or less the same except the graph convolutional networks. The one that was working relatively better was the autoregressor, these things I was telling you. So what we kind of show was that you don't need to go crazy with the partial differential equation or with epidemic spreading model, but you can use a simple multivariate autoregressor model and it's working, you getting the same results. Maybe with the graph convolutional neural networks, you will get the same results, but you will probably need more data. So it's complicated. It's science is not easy, and the good thing is that, as I was telling, that with either machine learning on these statistics, you can get more or less at the same point you will get with the partial differential equation or with some kind of epidemiology study. Another things that we tried recently also was still using these graph convolutional networks for epilepsy, where we substantially, this is actually something that got published this year. What we were doing was more like that this is it's how you will put your electrode if you do EEG, if you study EEG. Like you will put two here, like 4 elements here. Where you say Nasion is just selling the reference of the nose. And like this A1 and A2 are just the clip you will put in your ear just to have some bass. And what we had a lot of recordings from EEG. So this is not magnetic resonance imaging. And we were studying this again. We were comparing, trying to see if we were able to detect epileptic seizures. Why we can detect epileptic seizures is the reason is because yeah, as I was saying, we recorded time series and we were doing functional connectivity, as I was telling you before. But people who suffer from epilepsy, before they have the epilepsy, they have a moment that is called pre-ictal, where they are still okay, but at there are some subtle changes in their brain signal. And this is when we can actually predict that it's happening after 10 minutes a seizure. So that's why we are able to do this prediction. And in an ideal world, we imagine that this is someone wearing these things is driving. If it is some kind of, if he gets some kind of warning that if you get some kind of warning that is having in 10 minutes a seizure, is parking the car or is like sitting or doing something. Otherwise, if he has a seizure while he's driving, is maybe having an accident. why the important thing that we did it was also using something called graph attention networks. So while we were training the machine learning graph convolutional network, we were considering something that is called trying to give some kind of weights to the networks in the brain. Again, networks in the brain constructed according to the EEG and according to correlation in time, like I show you at the beginning. And we were trying to find the most, like, also because we were interested to understand which connection was important. And actually it worked very well. So we used the classification was kind of high. So the R under the curve was very high. So the accuracy was like 91% or around 91%. And this here you see the attention coefficient. So we found some coefficient that were particularly important in the in between this connection in the EEG. We haven't published yet, but we have some data from Nigeria, from not this nice EEG that we try here, but something smaller that actually looks more futuristic. Like, is this device here that you see? That's called that's called emotive. And we like so we these are not data acquired by us, but somebody else acquired, but they gave us. So they went in some rural areas and they came back and recorded from some subjects the same thing. And we haven't published yet, but it's more or less we did it the same thing with graph convolutional networks and the attention networks, and we were kind of also able to have a decent accuracy. I don't have the exact numbers because this is still something happening right now, but it worked well even with this small portable EEG in some rural areas in Nigeria. Then maybe some last things, and then I move to the business like. So the reason why I have a complicated story. So the story that I have to tell you is that sometimes when surgeons do for brain tumor, they need to cut some parts. And let's say what they do is that, of course, they put some injection in the subject, and then they see where is the most active part they have to remove, and they remove it. The problem is that when they do so, and for example, they have to cut this entire part. This part is not just the guy is losing part of the brain, then they are also damaging some parts of the language and movements, maybe. So what's happening is essentially that not only this person then is have to undergo this heavy surgery, which is very delicate, but then also after the surgery, this person maybe is not able to speak or to move properly. So what we wanted to do is that we were given some the initial structural connectivity, as I'll show you at the beginning. So this one that you see here is the adjacency matrix of the connectome I was showing you. So what does it mean is that each row is one region and each column is one region. And if one region is connected to another region, you have more value. It's another way of representing this connectome. So what we did it was that we had some connectomes of people with brain tumor before and after surgery, and we train a machine learning model to see if we can predict how it looked like. The reason why we are doing this is because if we can predict how it looked like, we can tell the doctor before he's doing the surgery how it will look like. So he knows he has to be more delicate, otherwise he might gonna cut some parts related to the language. So I don't know if it was clear the reasoning, but the idea was that we were able to predict what's going to happen after surgery, and we were telling it to the doctors, so they were doing better surgical planning. And we tried many things, from some complicated graph convolutional networks to even just simpler linear regressors. Then, actually, telling you the truth, the linear regressors were actually working good. It was not necessary to go crazy, because again, these graph convolutional networks, they were working well in theory, but then in the practical things, when you go and talk to do things that are used by the doctors, maybe even the simpler model is better. Now, why I'm telling you also this case is not just because I want to show off this story, but because you probably have seen that now there are a lot of people interested in let's say these VR things from from Apple Vision Pro, all these things, and yeah, what we investigated is that if we can give this to doctors in a way that they have it either in the with VR set, or they have it on their phone, so they have some kind of augmented reality and they can see these things I was telling you of the connectome or this graph. So and they also can see the predicted via machine learning model, so that they can make better planning. This is still ongoing. We are just started now the test. So it's another thing that we don't have it published. We don't have number, but we are still doing right now. I can show you how it looks like on a fake patient, because this is something we have here in the lab. So you see, okay, the big head is a fake head that exists in reality, and this that you see is the screenshot from like a smartphone. So what we have to do is that we need first to see here to adapt the fake head to the real head. So you see they are not really adapted, so we have to adjust it a bit. And once is once they are matching, like you see now that we are moving around, then we can start visualizing all these things I was telling you. Now here we're still playing with it, but I think it stopped. Okay, now we're like visualizing everything, so we can visualize where is the brain, where is this like tracks that we are interested, the connectome. So the connectome before surgery, we can also visualize the connectome after surgery. These things that I was telling you that we predict with some machine learning or whatever linear regressor, we can show you, look, according to machine learning, will look like this. And yeah, and the idea is that substantially before they do the surgery, they can see these things. Like you are seeing here with the fake head, but we are supposed to test this with the real people. So that is something we are doing right now. Now, why I told you this story is because what happened is that the many people here thought of this thing is so cool, we can not just write the paper, but maybe make a product and sell it, which seems a good idea, but actually is also a bad idea. And the reason why is a bad idea, because and as long as you do research or you do things with the hospital, maybe it's a good thing. But if you want to make a business, it's probably not a good idea, in the sense that these things might be very complicated. You might need medical certification, you might need this FDA, you might need patent, and then maybe it will be so complicated or so expensive that nobody will buy it. So it's not if we are still talking about science, these things are so cool, fantastic. All these things I told you, partial differential equation, outer regress graph, these things are cool. But if you want to go make a business, it's probably not the best idea. because in fact, one of the reasons why some startups anywhere, can be Europe, US, Africa, everywhere, they fail is because there is no need. So you make something fancy with something crazy, but then nobody's buying it. What instead you should do is the opposite things. Like this is something called design thinking, is that you should start from the need. Let's say I don't know, you go to some local clinic and you ask them, what is the problem you have, and they will tell you. Maybe I'm spending too much time doing the screening of for malaria. Do you have something that go faster than that? So that they will tell you the need, and then maybe that's the thing you should work on with your machine learning, not the other way around. So not doing something crazy fancy and then try to sell it, but go first to the doctor and ask them where's their problem, and then they will tell you and then maybe then you go back and think then maybe if their problem is that they spend too much time with doing malaria screening, maybe if I have something with that is doing some machine learning, analyzing the pictures of malaria, and is going faster, they will like it and they will buy it. So what I advise you is that one thing is that you try to do research paper, math, or you do something cool. But if you are want to make a company, it's probably better that you start going to the people and asking what they need. So that closing, because I think I really spoke a lot, is that like when we do research, we do many things, but also remember that one thing is doing research, that translation into clinical practice is complicated, and also a part that is complicated, not necessarily then you will be able to make a product that can be, because you have regulatory things, patent, you have to show that it's working. It might take you forever, and maybe mm, is not the right way to make a business. So if you want to be science, do science, but if business is probably better, you go down to earth and ask people. Maybe they just want something else, or maybe you end up not even in medicine, you do something else. I don't know, an app connecting people that wants a tractor, or selling something. Maybe these things is more so you should start asking more what are the need of the people, and so that don't be so arrogant that we are scientists and we also good at business. So maybe if you want to make this transition, it's probably better that you start thinking from what is the need, what do they need people, and then I define my machine learning things and so on. The good thing is that I've seen a lot of companies, even especially in Ghana and Nigeria, there are a lot of SAS companies, like software as a service companies. So there are many people doing stuff. There are many ventures even in Ghana. So it's a good. I would say there are many possibilities. Don't be too much stuck. If you want to go in this direction, don't be too much stuck with all these crazy things I show you, because maybe they are very fancy and cool, but for business, not much. And then, yeah, as I was telling you, I wrote a book with actually I put together all my experience I have in this. So if you want to know more in details, and like even people that are friends in Cameroon doing, they like it. So we had a lot of. If you want to, you can find it. Maybe I can send you one copy to the university. And then, yeah, we are looking for people at the moment. Though I don't have money. But maybe in 1 year we have more money and we can hire people. Although I know it's insane to get a visa to Poland from Ghana, but now that we know, we can try something. But don't write me right now. You can contact me maybe 1 year, because in 1 year we will have money and we can have more PhD students or scientists or something. Or if you have ideas how to collaborate or to do other projects, like feel free to contact us. I put here the address of the website. bam.sano.science, or my email is simply a.crimi@sano.science, or you can find me on Twitter or whatever. So I don't know. I assume you have some question for something, at least for the science part, or if you want, we can also talk about clinical translation. So I stop sharing and you let me know. I have another. I have another thing actually. I forgot it because I have the last advertisement. So we haven't advertised it yet, but there is a conference that is called Mickey, that is used to be always in Europe and US, but this year